Hello, good morning students. Now we'll discuss about immunoglobulin E. IgE is a class of antibody that has been found only in mammals. Only 0.05% of IgG concentration means it is present in the least amount. IgE exists as monomer consisting of two heavy chains and two light chains. The heavy chains, they are known as epsilon chain, and the heavy chain, it consists of one variable region known as VH and four constant region as CH1, CH2, CH3 and CH4. Ige main function is immunity to parasite such as parasitic worms like Schistosoma mansoni, Trichinella spiralis and Fasciola hepatica. Ige may also be important during immune defense against certain protozoan parasite such as Plasmodium falciparum. Ige also plays an essential role in type 1 hypersensitivity, which manifests various allergic diseases such as allergic asthma, allergic rhinitis, food allergy, and some type of chronic urticaria and atopic dermatitis. Ige also plays a pivotal role in allergic conditions such as anaphylactic reaction to certain drugs, bee stings, and antigen preparations used in specific desensitization immunotherapy. Ige was discovered in 1966 by Japanese scientist couple Tiroko and Kimishinge Ishizaka. So in this figure, you can see that on the mast cell, the Ige is present. The antigen, it is binding with the Ige and it stimulates the mast cell to secrete various chemicals like histamine, heparin, and can cause various responses. So mast cell, they releases the histamine as well as heparin, interleukin in response to Ige. So Ige is having receptors. It can bind with the receptors on the mast cell with the help of Fc receptors. The Fc receptors are also found on eosinophils, monocyte, macrophages, and platelets in the human. There are two types of Fc receptors. Fc epsilon R1, that is type 1 Fc epsilon receptor, the high affinity Ige receptor. Second, Fc epsilon R2, that is type 2 Fc epsilon receptor, also known as CD23, the low affinity Ige receptor. Fc epsilon R1 is expressed on mast cell, basophil, and the antigen presenting dendritic cells in the human. Binding of antigen to Ige already bound by the Fc epsilon R1 on mast cell causes cross-linking of bound antibody E and the aggregation of the underlying Fc epsilon R1, leading to degranulation and release of mediator from the cells. Basophils, upon the cross-linking of their surface Ige by antigen, release type 2 cytokines like interleukin 4, IL4 also known as, and interleukin 13 and other inflammatory mediator. The low affinity receptor, that is Fc epsilon R2, is always expressed on B cells, but its expression can be induced on the surface of macrophages, eosinophils, platelets, and some T cells by interleukin 4. So these are the two types of receptors present. Physiology. IgE may be beneficial in fighting gut parasite such as Schistosoma mansoni, but it has not been conclusively proven in humans. It is most likely benefit in removal of hookworms from the lungs. IgE may play important role in the immune system recognition of cancers. So, with the help of IgE, the killing of nematode larvae can be there with degranulation. IgE that can specifically recognize an antigen. Typically, this is a protein such as dust, mite, grass or ragweed pollen, etc., has a unique long-lived interaction with its high affinity receptor, that is Fc epsilon R1, so that basophil and mast cell capable of mediating inflammatory reaction becomes primed, ready to release chemicals like histamine, leukotrienes, and other interleukins. These chemicals cause many of the symptoms we associate with allergy, such as airway constriction in asthma, local inflammation in eczema, increased mucus secretion in allergic rhinitis, and increased vascular permeability. It is presumed to allow other immune cells to gain access to the tissues, but this can lead to potentially fatal drop in blood pressure as in anaphylaxis. So when the ragweed or pollen first is attached with the B cell, it leads to the production of IgE. When this IgE binds with the mast cell and second time the ragweed pollen attacks the cells, then with the help of cross-linking with IgE, it leads to the release of chemicals by degranulation and causing the symptoms of allergy. So, my sincere thanks to www.google.com for providing us beautiful pics so that we can explain the topic well. Thank you.